So at this point, I would like to introduce Anthony Ratcliffe. Anthony Ratcliffe, PhD, is the president and CEO of Synthasome, Inc., a biotechnology company specializing in orthopedic devices and orthobiologics. Tony obtained his Bachelor's of Science and PhD from University of Birmingham in the United Kingdom, and then performed postdoctoral research in London and was a faculty member at Columbia University. In 1996, he joined Advanced Tissue Sciences, where he served as Vice President for Research until 2002, when he founded Synthasome. Tony has chaired and served on multiple professional society, review, and editorial committees, is co-chairman of the Tissue Engineering Committee for ASTM, is a fellow of the American Institute for Medical and Biological Engineering, has published more than 100 papers, and is an inventor of multiple patents. Tony, with that, I'll turn it over to you.
Thank you, Kendra. I think we've lined this up very nicely. I'm going to give a general brief overview of my perspective on standards and their value in the field at this stage, and then focus on articular cartilage repair and how a standard process has been used to enhance that area of generating products to address that problem. In terms of looking at the opportunities for standards to impact the field of regenerative medicine, I've called out a few specific points here. One is that if you have a series of standards to be used, then you have the opportunity of improving the assessment of how much time it's going to take and how much it's going to cost. Now, that really has a big impact on being able to obtain resources, funding, for example, and also managing those resources. So right at the beginning of any program, standards can have a substantial impact. Now, as you move through the phases of research and development, particularly the early phases, once you have those standards, you know how much time it's going to take and how much it's going to cost. That means you can have increased efficiency in your process. And then as you move through, because you're working with standards, known methods and procedures, you have the increased opportunity to demonstrate effectiveness as well as safety. So this has the potential of enhancing your chance of success. Now, success, is that important? Of course it is. But particularly for the small business developing a new product, it is incredibly important that you avoid failure. A failed or poorly performed preclinical study or a clinical trial is often catastrophic. It really can kill off a program. And particularly repeating a clinical trial is usually not an option for a small business. Remember, small business, the startup companies are really where many, if not most of the products in this field will originate from. So it's absolutely critical that we optimize our opportunity for success. Standards can help us do that and really will help us avoid failure. Where do we use the standards? Well, we use the standards throughout the product development pathway, from the beginning to the end. And all of that lives within a quality system. The quality system you use will be a particular standard. Within that system, we've obviously got a various numbers of stages, from concept, feasibility, development, product performance, and all the way through to manufacture and launch. And we use standards in any and all of those different stages of product development. What we're going to do today is focus on a set of standards related to articular cartilage, which would fall into stage 2, which is feasibility studies. But again, standards are important all the way through, from the beginning through feasibility, clinical studies, manufacturing scale-up, and launch all the way through. In this particular example, we're going to discuss a standard that was developed at ASTM. So ASTM is a standards organization. There are a number of these, and it has an organization that provides the structure, resources, forum, technical expertise required to develop standards in a particular area, and also has the organization required to publish and disseminate those standards. The ASTM organization is set up as a set of technical committees. You have divisions, the divisions have subcommittees, and those subcommittees form task groups, and the task group is that organization that actually does the development of a particular standard. So this is the process that's used. There's a process that involves consensus. We have to build consensus throughout the assessment period. So all participants are represented, and their comments are assessed and evaluated. If they're found important, they're put into the standard. If they're found that it is not appropriate, then they're voted on and excluded. So there's a whole process there that involves everybody, and we have representation from industry, academia, and the regulatory bodies, both within the United States and internationally. Within ASTM, we have a particular set of subcommittees focused on tissue engineered medical products, TEMPs. And in there, we've developed over 40 standards related to tissue engineering regenerative medicine. Approximately 30% of those standards have been recognized by CDRH at FDA. We've got, at the moment, more than 10 standards in various stages of development. And we have participation in developing those standards by representatives worldwide. So ASTM is a nicely set up organization to develop standards in this particular field. The example we're going to talk about is osteoarthritis and articular cartilage. Now, OA and diseases of articular cartilage are common. They affect multiple joints. They have a significant impact on quality of life and productivity of the individual. The risk factors associated with OA and articular cartilage defects include age, obesity, and trauma. So within that patient population, there's a substantial number of patients, proportion of patients that are affected. Over the years, there have been only a few cartilage repair methods or products that have been marketed, particularly in the United States. Now, this area, we've chosen to exclude joint replacement. Here we're talking about articular cartilage repair. A little bit of the history here, osteochondral allografts, so were introduced in the late 1800s. So here we're taking another person's articular cartilage and subchondral bone and implanting that into the defect site of a patient. There was really little advance until the 1980s when microfracture was popularized by Richard Stedman. And here what you do is you have a defect site, you clean it up in the articular cartilage, you create microfractures that go through the subchondral bone of the defect site, and that creates a fibrous repair. And that has set a new benchmark for articular cartilage repair. In the 1990s, mosaicplasty and oats was developed. Here we're harvesting osteochondral plugs from a particular patient and then placing them into the defect site. in '97, the first cell-based product was introduced, and this is an autologous chondrocyte transplantation. Cartilage is harvested from the patient. The cells are isolated, they're expanded in culture, and then at a later date placed back into the articular defect site covered with a membrane, and this generates a repair. In 2007, another 10 years time span, and de novo NT was developed by ISTO in collaboration with Zimmer, now Zimmer Biomet, and this is an allograft of articular cartilage pieces harvested from young patients and implanted into the defect site. And then just this year, as was just mentioned, MACI was passed through the FDA process and is now on the market. This is an update of a Carticel, the autologous chondrocyte transplantation, and this involves, again, harvesting the patient's chondrocytes. They're then placed onto a membrane, and that membrane with the cells is implanted into the defect site. So what we got here is a combination of allografts and autografts and extremely slow advancement of new products through the market. So we really do need some help in bringing new products through. How does it look at the moment? Well, there's lots of interest and activity. If you search clinicaltrials.gov for osteoarthritis, you'll find there's almost 1,000 clinical trials in the US and approaching 3,000 clinical trials worldwide. If you search for articular cartilage, there's 39 clinical trials in the US and 132 clinical trials worldwide. So we've got a lot of activity. We would really like to see some of this come to fruition. But we face significant challenges, of course. How do we assess these new technologies? How are these products evaluated and compared? And how are they assessed for regulatory approval? So we've got some real challenges in dealing with this. At ASTM in the early 2000s, we recognized some of these challenges and identified the preclinical model of articular cartilage repair as an area where there was a substantial need, an opportunity to contribute to the field. And so we developed a standard guide for the in vivo assessment of implantable devices intended for repair or regenerate articular cartilage. So this is a standard guide is a standard that's intended to be somewhat generic and inclusive. And so that's the way it was developed. And it provided recommendations for animal model development and cartilage repair evaluation. Let's look at some of the points that were put in here. One of the things we included in this standard, which was developed again in a consensus manner with multiple levels of expertise. It included Richard Coots, who's an orthopedic surgeon, Barbara Boyan, an academic orthopedic researcher, Warren Haggard, who was industry R&D, and as well as myself, and lots of input from that type of broad expertise. We looked at the species, the different types of models that are being used. We considered all of the major ones, and as you can see here in this table from the standard, you can see we included rabbit, dog, pig, goat, sheep, horse. Those animal models were discussed in terms of joint size and load, how you handle the animals, affecting gender, age, study duration, and considerations specific to each species. We discussed the considerations for the defect site, whether it be the femoral condyle, trochlea, or elsewhere, what type of defect you might create, how the product might be fixed, whether you'll use joint immobilization. Then you move into the test procedures, evaluations and results, and statistical analysis. All of these things were considered, and it's been a particularly productive standard guide. Now, in 2011, the FDA published a guidance for industry on preparation of IDEs and INDs related to products intended for repair or replacement of articular cartilage of the knee. Now, this is a complicated issue, and this guidance was designed to be broad. It included description of the product, discussion of manufacturing, points about biocompatibility, etc., and clinical study protocols. Importantly, for this discussion, it also included a section on non-clinical data, including animal studies. Now, in that discussion, they had a summary provided, and it gave a nice overall summary. Most important for us, it made the following statement. It said, because a recommendation for a set of specific evaluations is not possible without detailed description of the articular cartilage repair or replacement product, reference is made to the ASTM F2451 standard guide for in vivo assessment of implantable devices. They state that this standard provides guidelines related to the development of animal models and mechanical testing, and they recommended that the applicants consult this standard. Now, we were, of course, very pleased that the FDA provided that recommendation, and it meant that there was an excellent communication between the team that developed the ASTM standard and those at the FDA. So there's a nice, smooth transition, mutually supportive here, of a standard having an impact on a guidance issued by the FDA. And we'd like to believe that both are being used actively for the development of new products addressing articular cartilage repair. Now, what we also recognized was that there was no specific standard model for articular cartilage repair. It still left major decisions to be made by the individual investigators. And now those decisions, in large part, would be dependent on site, species, type of defect, duration of the study, as examples. So what we've done since then is we've initiated the development of a standard test method for testing articular cartilage repair or regeneration. And this is an ongoing work item at ASTM. And this work was funded by NIH. And here we selected from the ASTM standard a GOAT model for osteochondral repair. And this standard has a very specific experimental design with appropriate controls. It defines duration. It is going to describe the results and therefore the expected outcome, in particular the controls. And it will therefore permit efficient experimental design planning, which will of course include costs and time. So we now are seeing a pathway where you start off with a standard guide, moves through the FDA guidance document, and down to a very specific standard test method. So we've got a very efficient system being described and how to perform an animal study that would be appropriate to assess new technologies for articular cartilage repair, the time and funds required, the experimental design, and all associated analytical methods are all discussed between the three different documents. So as a quick summary, we can see that what we have is that the standards have provided a rational approach for us to assess and compare different technologies. They can substantially increase efficiency of product development and regulatory submissions, and it requires the participation of any and all stakeholders to develop a consensus. And this last part is particularly important. We really need participation of all the stakeholders to ensure that the patents that the standards that we develop are as effective as they possibly can be. And I think I'll stop there. Thank you.
Thank you very much, Tony, for that history as well. And we've set some time aside for some questions for you. For the group, as a reminder, if you have questions, please enter those into the chat function by clicking on the question mark icon to the right of your screen. This may be a question mark or a circle with a question mark inside of it. So I'm going to give people a little time to enter additional questions. But in the meantime, Tony, you provided a great history lesson on the articular cartilage repair standards development process. I note the timeline for this work spanned decades. Do you think that this is the norm, or as the entire regenerative medicine field advances rapidly, can timelines be accelerated?
Certainly the expectation is that if we know what standards we can use, we'll be able to define the product development pathway much more clearly, and we will have a much better level of expectation in terms of success or, of course, failure. But at least we'll know what we have to achieve, and so the target is clear. We don't know what to achieve, and we have to figure that out as we go along. There's a vast amount of uncertainty. So in articular cartilage repair or any aspect of regenerative medicine, if we know what our targets are, then we can go away and achieve them. Achieving a moving target is really, really difficult.
So reducing that uncertainty makes a big difference. What do you feel should be the community's goal for moving from a recognized need for a standard to when a standard is available?
Well, the community has to participate. It's all very well to wait for a standard to arrive. If the community does that, then of course they don't have much input, and it may not be ideal, at least from their point of view. So the best thing for them to do is to participate in developing standards. That way they can not only ensure that voices particular standards, but they can have standards developed that they care about. And so there's not only the standard, but prioritization there. So for both reasons, the community has to participate.
Okay. And then another question we had is, looking back on the experience with articular cartilage standards, what would you do differently in hindsight to make the process more efficient or the results of the process more effective or useful? And kind of, are there any lessons learned you'd share with the group or mistakes that you'd recommend they avoid?
The challenge, like many challenges in R&D, is not only developing the standard, but here the articular cartilage repair standard is very expensive to develop. And so what is important is if you're going to develop the standard, recognize that resources are required. And the progress of developing a standard is dependent on the availability of those resources. So please note that effort and resources are required to put these standards in place. And that's something that we continue to learn and recognize. And so if I've learned something here, it's that you should not underestimate how much it costs to get these things done.
So understanding it and planning for it sounds like it's really important. A final question is, how can SCB's efforts complement ASTM standards development from your perspective?
Well, a couple of areas there quite clearly in my mind. One is that they can identify and prioritize standards that need to be developed. And also, too, they have the capability to provide the resources. And by resources, I mean in particular the people, because you have recognized experts within that group, and that membership will only continue to grow. So being able to identify people, experts that are able to contribute would be a really substantial addition to the field.
Well, thank you very much for offering your perspectives, the history, and your experiences to this group overall. I appreciate you taking the time, Tony. Very much appreciate it.